All right, folks, so now we're on lecture 25. Now, I would mention that after lecture 24, you might be a little bit confused as to how a strong input that we saw in associative learning, like processing pain from a foot shock, might then sustain a weak synapse associated with something like processing a very simple tone. So that figure might have been a little bit confusing. As always, feel free to ask me any questions that you have. Hopefully, what we talk about a little bit today will help clarify some of the issues about how that strong synapse can enhance a weak synapse in associative learning. We are going to talk about the different parts of the amygdala, and hopefully that'll make a little bit more sense because the amygdala has a lot of inputs and outputs to different parts of the brain. But before we get that, we're going to talk a little bit about some of the different types of learning. So now that we understand the basics of that proposed cellular mechanism for learning, long-term potentiation, we can now talk about some of the different types of learning. Now, something that you should probably keep in mind is that we don't know whether long-term potentiation is actually necessary for all of the different types of learning that there are. Now, although long-term potentiation has been studied primarily in the hippocampus, it does occur elsewhere in the brain, like the amygdala, and this could support other types of learning that do not require the hippocampus to be able to do. So one type of learning that we are going to talk about, is perceptual learning. So perceptual learning is content specific. It's often involved in learning different types of stimuli, learning somebody's face, learning words, learning what different sounds are, learning different types of textures. And these contents are typically processed by somewhat distinct brain areas. So if I'm processing sound, it goes without saying that regions of the temporal lobe and the auditory cortex might be involved. For faces, obviously things like the fusiform gyrus. For different types of textures, areas that would be important for processing touch will be involved. So oftentimes, especially for a lot of knowing things, especially those things that have a visual component, we do know that the what pathway is also very involved for visual stimuli. So for example, the fusiform gyrus in the inferior temporal lobe is really critical for faces. If we're looking at scenes, the parahippocampal gyrus, also in the inferior temporal lobe, is also involved. Now, interestingly enough, for those who are kind of curious about language, the left inferior frontal gyrus, kind of around Broca's area, but encompassing a lot of other areas as well, responds to both auditory and written words. And we know from patient lesions and intracranial recording studies that these content-specific areas are really important for perceptual learning and memory for these different types of contents. So for example, we have an area right here. So if I show you a face, we get a lot of activation in the fusiform gyrus. Interestingly enough, there is believed to be a specific body area that is very highly activated when we are looking at different types of bodies. Now, if we're looking at these areas, you can kind of see that they are, we do have some areas that are functionally distinct. This is based off of neuroimaging studies in healthy young adults. And so it has been suggested that we do have areas in our brain that have neurons that are very strongly specialized for the learning and the memory of faces and bodies, etc. So that's perceptual learning. Now, let's talk about associative learning. And I briefly touched on classical conditioning on the last lecture, but I thought it would be worthwhile to go over it a little bit again. So classical conditioning occurs when an unimportant stimulus that typically does not produce a reflexive response and wouldn't under normal circumstances basically takes on the properties of an important reflexive stimulus when those items are paired together. And so this was something that was originally discovered by the physiologist Pavlov. And so how we tend to talk about classical conditioning is as follows. So we have an unconditioned stimulus. This is something that produces a reflexive instinctual behavior that is not learned. So for example, in the famous Pavlov study, if I put food in a dog's mouth, it will begin to salivate as a response. Now, the dog did not need to be trained to salivate to food. This is something that the dog does reflexively. So for this reason, we often call food, in this case, an unconditioned stimulus, and we call salivation in response to that food our unconditioned response. Now, our conditioned stimulus is basically a neutral, unimportant stimulus that normally does nothing on its own, like the sound of a bell. And when we pair that with our unconditioned stimulus, begins to take on the unconditioned response, which we call the conditioned response. So in Pavlov's study, for example, we pair the sound of a bell with food. Now, here's what's really critical. The bell has to come before the food because the bell is supposed to be predictive. The bell lets the dog know that food will soon arrive. So timing is absolutely critical for this. So we repeatedly present the bell with the food, the bell with the food, the bell with the food. And eventually what we will end up seeing is that the dog will begin to salivate to the sound of the bell before the food is even presented. So the dog did not need to learn to salivate in response to food, but it did have to learn to salivate in response to this bell. And for this reason, we call salivation to the bell our conditioned response. So as I talked about in the last lecture, another way of thinking about something being conditioned is that it is learned. So this is a type of learning where associative LTP may be important. Now, operant conditioning is sometimes referred to as instrumental conditioning. This is a case where a behavior is modified by the consequences of that behavior. So operant conditioning does not involve reflexive behaviors. It involves a learned behavior in association with some type of consequence, and it can be modulated by either reward or punishment. So for example, we can teach a rat that if it presses a bar, so if we have a stimulus like a lever that the rat can push on, if the rat gets food as a reward for pushing the bar, we will begin to see that behavior increase because the behavior is being reinforced, which means it will strengthen the behavior. So here are a couple of different examples of associative learning. So again, we have our fear conditioning that we talked about in lecture 24. This is very much a type of classical conditioning. It is sometimes referred to as conditioned suppression. And there is some evidence, as we saw last time, that long-term potentiation in the amygdala is very strongly correlated with the freezing response in a rat. Now, interestingly enough, areas that you might predict would be involved in this conditioning are not necessary for fear conditioning. So you might think that the hippocampus is really important for learning, or areas like the cerebellum and the sensory cortex. And in fact, all of these areas are critical for learning different things. So the sensory cortex may help us process pain that we might experience from a foot shock. The cerebellum could be involved in learning the timing between the tone and the foot shock that later pops up. The hippocampus can help us remember that we got a foot shock. What's really interesting is that you would think that all of these areas are involved, but it turns out that when researchers lesion the somatosensory cortex or the cerebellum or the hippocampus, you still get fear conditioning. So these areas might be helpful for fear conditioning, but they are not necessarily sufficient, nor are they critical for fear conditioning. On the other hand, though, we know that the amygdala is critical for fear conditioning. We know this because if you damage the amygdala, fear conditioning no longer happens. So regions that you might predict based on other types of conditioning are not necessary for fear conditioning. Now, you might be kind of curious. How can fear conditioning occur after damage to the primary somatosensory cortex? If you're getting a foot shock and you have damage to the somatosensory cortex, you wouldn't be able to process it, right? So how can this happen since you can't process a shock? And it turns out that the amygdala is involved with a lot of different areas of the brain. And the amygdala is really essential for decoding emotions, fear in particular, and many sensory inputs help inform us of potential dangers of the environment. So here's what's really critical. This sensory information can either come from the thalamus, so the sensory portions of the thalamus, or it can come from the various sensory cortices. So that would include things like the visual cortex. That would include things like the auditory cortex or the somatosensory cortex. But in addition to these areas, it also gets inputs from the hypothalamus, the septum, and the reticular formation of the brainstem. Now, here's what's really important. Let's say we've abolished the sensory cortex. There are still other regions that might be processing aspects of the foot shock, even though you can't actually process the foot shock. So remember that this sensory information, before it ever goes to the cortex, will go to the thalamus. So some aspects of the thalamus are still processing the foot shock, even if the sensory cortex can't. And so because of that, generally what we will find is that if we knock out one of these potential areas, like the sensory cortex, there are all of these other areas that are processing information in the environment and are still sending their activation to the amygdala to create an emotional response. So if you knock this out, it doesn't really matter all that much because all of these areas are still having connections. So this is something really important to know. Additionally, the connections between the amygdala and the hippocampus may actually be the source of emotions that may be triggered by certain types of memories, particularly those for traumatic events. Now let's talk about the different components of the amygdala. So, like the basal ganglia, it's composed of several different nuclei. So we have our lateral nucleus right here. And you will also notice that we have an aversive stimulus here that creates that strong synapse. And that's because areas like the sensory cortex and the thalamus are often processing those strong reflexive inputs. So if I get a foot shock, that is going to create a strong synapse right here. But notice also that different sensory cortices, like the auditory cortex and the visual cortex and the somatosensory cortex, are also involved. So here's my aversive stimulus, my foot shock from the somatosensory cortex giving input to the amygdala. And here is the auditory cortex providing information about the tone. Now notice that they're converging on the same input in the lateral nucleus of the amygdala. So here's our strong input, here's our weak input, and because they converge on the same inputs, because of that, this strong synapse can create a connection and enhance the weak synapse. And so this synapse is actually strengthened by this pairing. And it's believed that the lateral nucleus right here is where the conditioned stimulus and the unconditioned stimulus inputs basically converge, and we get that associative LTP. So the unconditioned stimulus, in this case the foot shock, strengthens the synapses with the conditioned stimulus inputs with the tone. And the outputs, That includes outputs. So the lateral nucleus projects to areas like the central nucleus, the basal nucleus, and the accessory basal nucleus. Once these all travel to the central nucleus, we get these different behavioral responses. Outputs are sent to the hypothalamus, the midbrain, the pons, and the medulla that basically mediates the different things that we see when we're afraid. It mediates freezing behavior. It mediates increased heart rate, dilation of the airways, and dilated pupils. So this is part of the reason why we believe that associative LTP occurs in the amygdala, because these inputs converge in the same portion of amygdala. So that's what I wanted to talk about with classical conditioning. Now let's talk a little bit about operant conditioning. So this is when you learn an association between a behavior and its consequences. So part of the reason that we call it operant conditioning is that classical conditioning, by and large, involves a reflexive behavior that is being conditioned. For learning to occur, you don't actively have to do anything. It's very, very passive in nature. However, if you want to learn anything with operant conditioning, you actively have to operate on the environment and do this voluntary behavior so that you can learn the consequences. So instrumental is voluntary control. That's why we call it operant conditioning. You are operating on the environment. So for example, if you get to work on time, you don't get fired. And so you might have, and for you to make that connection, you actively have to do something. And this can be modified or modulated by reward or punishment. Generally, reward increases a particular behavior and punishment will typically decrease a behavior. So a very famous study that kind of looked at this, we typically think about B.F. Skinner when we think about operant conditioning, but the first person to really look at this was Edward Thorndike. And so what he actually did was he put cats in a series of these different puzzle boxes. And these puzzle boxes had pulley systems and platforms and different types of levers that the cat would have to engage in to be able to escape. And here's what's really critical. In addition to this pulley and lever system, we do also have a plate of fish or a food reward for the cat when it escapes. Now, the first time that the cat escapes from this puzzle box, it's typically done randomly. The cat is basically trying anything it can to escape from the box. And so because of that, what we often tend to find is that that first escape is accidental. But when you keep putting the cat in the box over and over and over again, especially if there's that food reward for escaping, what we tend to find is that the cat gets much, much better at escaping from the box. It escapes more quickly and it makes fewer mistakes. And so it's suggested that the cat eventually learns that pressing on the lever results in being able to get out of the box and get the food. And so Thorndike actually kind of termed this something called the law of effect. Basically, what the law of effect says is that behaviors that result in positive outcomes or valued outcomes will increase that behavior. So in this case, if the cat escapes, it gets food. Generally, I think most cats like food. And so that's very rewarding for the cat, and that increases the likelihood that the cat will escape in the future. Now, on the other hand, behaviors that are followed by negative consequences will tend to be less likely to be followed. And that's kind of what the law of effect is. And it basically tells us that if we reinforce a behavior, we're going to see that behavior repeated. If we punish a behavior, we're going to be less likely to see that behavior. Now, typically, the more that this pairing occurs, the stronger the response will be. And it will be faster and without error. So here's what this might potentially look like if we were dealing with a rat learning to press a bar and receiving food for it. So here we have a particular stimulus. This is actually seeing a lever. This will send information to a perceptual system. In this case, it would probably be the visual system. You're trying to see the lever. At this point, what you're going to find is that the rat will learn to engage its motor system to produce a given type of behavior. Now, here's what's really critical. So we have our perceptual system, our motor system, and then the rat gets reinforced for pressing the bar. Now, what system or pathway have we talked about that is really critical for pleasurable or rewarding aspects? The mesolimbic pathway, of course. And so that's that dopaminergic pathway that starts in the ventral tegmental area and terminates in the nucleus accumbens and makes use of the limbic system. So what we're going to find is that this mesolimbic pathway and processing this reward may potentially strengthen the connection between the perceptual system that codes for the lever and the motor system that controls that behavior. So this is just one way that associative LTP could potentially work in operant conditioning. So let's talk about a few different types of classical preparations and different types of conditioning preparations. So eye blink conditioning is a very familiar type of classical conditioning. I'm sure those of you that wear glasses or contacts have probably experienced this. When you go to the eye doctor, you get that wonderful glaucoma test. So basically what you do, for those who don't have glasses or have been lucky enough to not visit an eye doctor, what they will do, they'll kind of sit you in a chair and they will shoot a puff of air into your eye. Nobody really likes the puff of air except for me. It makes me giggle. It's really weird. I don't understand it. But you can utilize this instinctual reflexive behavior in classical conditioning. So a puff of air to the eye elicits a blink. Your eye instinctively wants to protect itself. So the puff of air is our unconditioned stimulus, and the eye blink is our unconditioned response. Now, what we can do is, prior to that puff of air, we can pair a tone with that puff. So in this case, the tone does nothing on its own, so it's going to function as our conditioned stimulus. Then, after repeatedly pairing our tone with our air puff, the tone will eventually elicit a blink, a conditioned response on its own. So blinking to the tone will be our conditioned response. Typically, this eye blink conditioning has been studied in rabbits. It's been used quite a bit to study eye blink conditioning and study classical conditioning in rabbits. This does occur in humans too, but obviously we're not necessarily going to lesion brain areas in humans to create this type of conditioning or to abolish it. So research with eye blink conditioning, basically we can look at two different types of conditioning in rabbits and whether or not memory is involved in this. So what we can do is either what we call trace conditioning or what we call delay conditioning. So in trace conditioning, we present our conditioned stimulus, our tone, and then after some period of stimulus-free delay, so notice that in this figure, no stimulus is being presented at this point. The tone goes beep, and then for a little while, there's nothing, and then finally you get the air puff. So this type of conditioning will probably require having a memory that the tone was presented if this type of conditioning is to occur. Now, in contrast, we have what is called delay conditioning. Now, in this particular case, we keep our tone on through the entire period until that air puff is presented. So we go beep, puff, like that. So there's no empty period here. The tone is on the entire time. And so odds are pretty good you don't necessarily need to have a memory of the tone. In this case, you might need to know how long the tone actually is. So looking at these two different types of conditioning, it's very clear that they require use of different types of brain areas. So trace conditioning, as I mentioned, requires having a memory of the conditioned stimulus. And so what this tells us is that this might require the hippocampus. The hippocampus can be really important for eye blink conditioning whenever there is a delay between the conditioned stimulus and our unconditioned stimulus. So this is hippocampus dependent. As we're going to learn very soon, the hippocampus is really critical for any type of memory that has to last over a long delay. And so if we actually lesion the hippocampus in rabbits, we do find that the rabbits are no longer capable of trace conditioning at this point. So this seems to be really critical for memory formation, particularly those memories that we need to retrieve after a delay. And we will learn soon that for many people that develop amnesia through things like stroke or heart attack or traumatic brain injury, one of the things that we will find is that oftentimes this amnesia results from hippocampal damage. Now, compare that with trace conditioning to delay conditioning. Now, because the tone and the air puff overlap, because the CS and the US overlap, memory here is not required. So what we tend to find is that if you lesion the hippocampus in a rabbit, delay conditioning still occurs. And we can actually find with amnesic patients that they also can still engage in delay conditioning. So the hippocampus is not critical for this type of learning. However, we do know that if you lesion the cerebellum in a rabbit, that this will negatively affect delay conditioning. And this is because the cerebellum is really important for coordinating the timing of movements, as well as keeping track of passing time, like a clock. And it's believed that this contributes to the eye blink reflex being initiated in response to the air puff. Interestingly enough, the cerebellum, like the hippocampus, also exhibits a lot of long-term potentiation and long-term depression. So we've talked about perceptual learning, we've talked about associative learning, and now we're briefly going to talk about relational learning. This last form of learning is definitely the most complex. So for example, if you look at, it's basically a formation of complex associations. So if you look at a family photo, it will often tend to trigger memory for multiple types of associations. And this is because it involves a very complex network of neural circuitry. So if we have associations that are potentially visual in nature, the visual areas will be activated. We might have activation of auditory areas, olfactory areas for smell, and so on. Now, because of the wide neural circuitry that is involved in forming and retrieving relational memories, many different types of disorders and brain damage can actually impair this type of learning and memory. So now we're going to kind of move on a little bit, and this is where we'll kind of wrap up for the week. We are going to start talking about some of the different types of memory. Generally, this will apply largely to humans because this is based off of the work by cognitive psychologists that usually tend to work with humans. But we do know that this is capable in certain types of animals as well. And these types of memory are supported by the different types of learning that we have already discussed. So what you're looking at here is a very, very basic model of memory. This is a model of memory that was originally developed by Atkinson and Shiffrin in 1968. It is sometimes called the information processing model of memory, or rather the modal model of memory. And so here's kind of how it works. So there's some type of stimulus out in the world, out in the environment, and we sense that through things like touch, through things like sight, and through things like hearing, our sensory organs. So some stimulus is processed by our sensory organs that creates some type of perception. Now, at this point, this is where sensory memory really comes into play. So sensory memory typically lasts between maybe a couple of 100 milliseconds to about 1 second. So sensory memory is designed to basically keep track of what is currently going on in your environment, and it's also meant to help you recognize change. These representations in sensory memory are really, really short. They don't last for very long once the stimulus is removed. So for example, if you've ever played with sparklers and you've made shapes with those sparklers, you'll notice that eventually once you make a shape, it will fade very, very rapidly. That is your visual sensory memory in action. Now, here's what's really critical. You need to be able to sense and sense what's going on in your environment. You need to keep track of what has changed in your environment. But only some of the things in your environment are important for you to remember later. So attention, very, very critical for getting information from sensory memory into our longer term types of memory. And so we pay attention to information in sensory memory, and at that point, this information now goes to what is called short-term memory, or some researchers call it working memory. So short-term memory, as the name would suggest, is incredibly short. It only really lasts less than a minute, maybe a little bit more if you rehearse things over and over again. So oftentimes what we will find with short-term memory is that it's very, very limited in nature. So oftentimes forgetting from the store can be really, really rapid. And so it's basically a short-term buffer. Information is not meant to stay in short-term memory forever. And so because of that, if it's information that you need for later, so if you're trying to remember somebody's phone number so that you can put it into something, what you're probably going to do is repeat over and over and over again. That can help put this into long-term memory. But what we find is that because short-term memory is so limited, oftentimes information that is dropped from short-term memory will be forgotten. Now, if we can repeat information enough over and over again, at that point, that information will go into long-term memory, where this information can last for days, months, or years. So really briefly, let's talk a little bit about short-term memory. So this is a very temporary form of retention. So memory is retained here until it's forgotten or until it's been moved into long-term memory. There are likely different types of short-term memory out there, possibly phonological for verbal materials, a visual short-term memory. And we do actually know that some patients do have intact short-term memory for words, but not for visual information. Now, this kind of memory can be affected by different types of neurological disorders, primarily for those affecting frontal and parietal regions. The capacity of short-term memory is incredibly limited. Originally, it was proposed that the number of items that you could hold were basically between 5 and 9. So it's often referred to as the magical number 7 plus or minus 2. So if I give you this string of letters, odds are pretty good you may only remember about 7 of them. But this is debated. If you take cognitive psychology, you might actually learn a little bit more about why we're starting to kind of move towards the idea that short-term memory is probably closer to about 4 items than about 7. And then finally, one of the ways that we can increase the information that we hold without overloading our short-term memory is through a process known as chunking, which was originally discussed by George Miller in 1956. So if I give you a string of letters like this, if you have pre-existing knowledge of acronyms, you can take this string of 9 letters and you can basically break them down into 3 different acronyms: IBM, CIA, and FBI. Now, some people think that working memory or short-term memory is separate from long-term memory. Some people just think that working memory is just activated long-term memory and possibly the same system. There's still a lot of debate on the relationship between short-term and long-term memory. Now, this is a slide I enjoy. So there are different types of long-term memory for the different things that we do. This is just a model. Everybody kind of has their own idea of how long-term memory is broken down, if it can be broken down into these types. So there's a lot of debate about these sort of distinctions. So we're going to make 2 major distinctions right now. So we have what's called declarative memory or explicit memory. This is dependent on relational learning. It is incredibly complex. We also have what is called non-declarative memory or implicit learning. And in these particular cases, they do not involve relational learning. Now, declarative memory can be broken down into memory for events in time and memory for facts, episodic memory and semantic memory. Now, some people believe that these are distinct and separate systems. Other people do not. So semantic would be, for example, what is the capital of Missouri? Well, it's Jefferson City. I don't remember where I first learned that, but I know that it's Jefferson City. Now, on the other hand, I can remember what I did yesterday when I took the dog for a walk. So there are some people who believe that there's no difference between these 2 types of memory. It's just the time factor. And it's possible that if we retrieve an episodic memory over and over and over again, this might actually become semantic over time. So declarative memories, we call them declarative because you demonstrate that you have this memory by telling somebody about it. You can verbalize it. Non-declarative memory is called non-declarative because you demonstrate that you have the memory by a change in behavior, and it's not necessarily something that is very conscious. So implicit is typically unconscious, explicit is often conscious. Non-declarative memory typically involves the forms of learning that we've already discussed. So that includes things like conditioning, perceptual learning, skill formation, and we will talk a little bit about priming next time. Now, as already discussed, for the non-declarative forms of memory, there are somewhat distinct brain regions that support these forms of learning, such that different types of patients exhibit different types of deficits. So some people that have impaired learning for faces, so a perceptual learning, they have prosopagnosia, do not have problems with fear conditioning. So these are very distinct brain areas supporting these processes. So that's what I've got for this week. We will talk some more about memory next time.